Tejah? Yes.
Rather, I should request Aditya to come forward.
Yes. Aditya, please come.
So it's really thanks to Aditya and Urvi and the other colleagues at Goethe-Zentrum that the exhibition is as pretty as you see around today.
Thank you.
And Amita for making this space for us.
Pleasure.
Thank you.
I have done.
So today's session is a collaboration between CCMB, Goethe-Zentrum and Sci-City. Sci-City is a collective that I, Tejah and other science enthusiasts in the city run, where our hope is to bring science into cultural conversations in the city. And I hope that today's session here with art and talking to scientists bring more inspiration towards that. A little bit about the exhibition, and Nikhil, if I may have the slide of the artists. The exhibition here, which is called Earth Million 2025, is designed by Orgho Manna and Suman Chaudhary. Both of them are artists in residence at IIT Kanpur. Orgho is a cancer researcher turned into a self-taught comics artist. He is very well known for his work in history of science done through the medium of comics. His work can be viewed at drawinghistoryofscience.com, which is his website. And he's been an MIT CAST fellow, where he worked a lot on how to talk about infectious diseases through the medium of comics. Suman, on the other hand, is a much more traditional painter, cartoonist and illustrator. He is a fellow of the Lalit Kala Academy and has taught at Sherwood College and NIT. They made this exhibition in collaboration with Co-Futures Group at the University of Oslo. As a part of the Co-Futures Group is Professor Bodhisattva Chattopadhyay and Professor Jeffrey Bowker from UC Irvine, who first proposed something called as the ant network theory, and they acronym it as again ANT, ant. And the exhibition here is based on this very theory. Ant is a thought experiment on the nature and evolution of non-human intelligence. It was originally published in 2019 as a short science fiction piece and manifesto in Science and Technology Studies by Chattopadhyay and Bowker in the journal called Nature Culture. It was then expanded and turned into a graphic novel by Orgho. The Earth Million 2025 exhibition is an excerpt from that novel that Orgho has made. It braids speculation about multi-species entanglements and post-species developments with the history of ants in global science fiction. The narrative here begins in a dystopian future in which humanity is in danger of being extinct. Orgho says the dystopian scenario first occurred to him a few years back because of the turmoil in many parts of the world caused due to war. He imagines that humans would not be good ancestors to the future life on Earth, and we would make every corner of the Earth ill-suited for life. The exhibition is divided into 7 stories. In the first story, Home in the Nuclear Winter, the majority of humanity and other known creatures go extinct as a result of a nuclear conflict. Few nomads who believe in collective intelligence and co-evolution survive. However, as we all know, life finds a way. In the following stories, he, however, imagines soil, air, water, and other elements that are required for life. New kinds of species appear. Orgho predicts super beings. These super beings are hybrids of multiple species, and he says that these would bypass the traditional order of speciation in terms of evolutionary success. He imagines a future without language and other cultural barriers. It will be the swarm intelligence that connects us all, as is the case with the ants, Orgho says. While this exhibition is speculative in nature, it is set on an Earth which is million years away from now, predicting the life forms that will emerge then. The discussion today, on the other hand, roots us to reality. It brings us back to understanding how new life forms really emerge. And to help us in that process is Dr. Jahnavi Joshi here, who's an evolutionary biologist. She leads a research group at CCMB. And she brings in a rich experience of studies from other premier institutions such as the IISc, NCBS, and the Natural History Museum of London. So in our session today, Jahnavi will first give a short talk where she sets context of what an evolutionary biologist's work really looks like, what do they do. And after that, she will be in conversation with Tejah Balantrapu, who is my fellow science communicator in Hyderabad. And however, Today, he will be playing a role of an active citizen who studies and works actively towards protection of biodiversity in the city. After that, the floor will open up for the audience to ask all your questions, and I hope that the conversations today will then set the context for you to navigate the exhibition that Orgo and Suman present for you. With no further ado, I'll invite upon Jahnavi to take stage.
Thank you so much, Somdatta, for that introduction, and first of all for inviting for Tejah and Somdatta to think about it, and Jarma Center and Amita to have this conversation here. You guys, so I have prepared few slides for 15 minutes or so to give you the context of the landscape we live and work in, which is peninsular India. And if you have questions during those 15 minutes or so, please feel free to ask me and stop me, and so that we can take those questions right there. But otherwise, afterwards is also fine. Just before we started talking here, I was going through the presentation here, and the context, as Somdatta correctly said, we study how the species we see around us today have come about. So the past of it, does that hold some answers for the future? We are still trying to see that, but our aim here is to see how the past processes or past geoclimatic processes have shaped the biodiversity we see today, and how, if any one of us want to study this, how will we go about it. And the landscape, as many of you are very familiar with, you live in Hyderabad, which is part of very much Deccan Peninsula, and that's the landscape today we will be talking about. Here's an image or a topographic image or a digital elevation map of peninsular India. Peninsular India in tropical Asia is one of the most ancient landmass on the earth. Why so? You can see, and it also hosts a global biodiversity hotspot, which means it has a high diversity, which is the western escarpment which you see on the west coast of India, known as the Western Ghats, along with the Sri Lanka. On the east coast here, you have much more broken chain of mountains known as the Eastern Ghats, and in between what you have is a vast expanse of Deccan Plateau, which has thorny scrub forests, grasslands, moist forest, and on the north you have Vindhyas and Satpuras. So if you think about it as one landscape or a one island, what happens? How does biodiversity evolves in this kind of a landscape? Just to give you another perspective of the same landscape, this is the Deccan Plateau, the Western Ghats, the Eastern Ghats, and the other mountain ranges. Has it always been like this, the current configuration which we are very used to seeing it? No. Many of you may be aware of the theory of continental drift. I am going to play a small clip here now. Can you assist me with the clip? Wagner proposed this continental drift theory in early 19th century. He didn't receive much support at that point of time, but in few years it became very apparent that Pangea, that today the continents the way we see them were as one landmass, and now what this clip is going to show you is that they are breaking away. You can see, keep focus on this landmass. You have South America, Africa, Antarctica, Australia, and this is peninsular India. Throughout its evolutionary time scale, how has it evolved? And now it's going to travel north. It's separated from Africa, now it's separated from Madagascar. You can see some small landmasses, chunks which are appearing and then disappearing. This is one of the landmass on the earth which has traveled the most in the sea. And then collided with Asian plate, Himalayas came up, which also set up the monsoon which we have. It also set the climate the way peninsular India has today. And now this is the current configuration as we see the earth, right? But if you have such a complex geological blast, it's bound to influence what biodiversity you see today, right? So just to give you a glimpse of it, it was peninsular India was part of this Gondwana landmass. It broke away, the breaking started around 200 million years ago, other landmasses kept moving. Final collision, and geologists debate on it, was it 45, 50, 40, because first it was a docking, then final collision, and then Himalayas came about. Okay, so it was an insular landmass which traveled in the sea, so you can think of it as an island actually moving at a very small or a slow pace. Another very important thing which happened to this landmass was a massive volcanic activity, and which was happening around the time India, peninsular Indian plate is what geologists would refer, was moving along the reunion hotspot, and that caused a massive volcanic eruption. We understand now it wasn't a sudden event, it went on for few millions. That changed the climate of the landscape. Here is the extent which is based on the fossil foraminifera, how and what portion of the peninsular India was affected by it. Certain areas acted as a refuge where it didn't affect as much. The climate changed but not as much as certain areas. As we mentioned, the Indian plate kept moving, finally collided with Asia, but when you start and your starting point and the travelling distance, you are passing through an equator. That changed the climate. It used to be really wet per humid climate, then it became seasonal after it collided with Asia. Why? Monsoons were set up. You had now sudden huge mountain range which is going to block the way the winds come from. And the monsoon as we see today in Indian subcontinent was set when Himalayas came up. They were intensified later, around 10 to 12 million years. There were cycles of aridification, monsoon intensification, and the last we know is global, like last glacial maxima. So given this is the geological past where it's not only the plate, its position has moved, its climate has moved, and of course that may have influenced the kind of habitat it has. What we see today is this is the topography, and this is just to give you a context when we are going to think about evolution on this place. This was not a constant plate on which this evolution was happening. The climate was changing, the geographic position of the plate was changing. Now what we see it today. How we see it today is this. You may have to bit reorient yourself. This is same peninsular Indian plate. This is Sri Lanka. This is Kerala, Karnataka, Maharashtra, Gujarat. Right? This is the mountain change known as Western Ghats. You can see this is the elevation, how the Western Ghats are. You see that there are taller mountains in the south as compared to the north. You have mountains going from 100 to 2500 meters in the south. As one moves towards north, it's around 1000-1200. That's your Eastern Ghats, starting from Orissa, Andhra, Telangana to Tamil Nadu. All of this is in the middle, which is Peninsula or Deccan Peninsula. We are somewhere here in Hyderabad at 700 meter elevation. Why is this elevation is also important? We'll talk about it in a minute. Also, the monsoon enters from the south and retreats in the north. So the rainfall will also change, but what is interesting and important here, which is the seasonality. The amount of rainfall is important, but how long does it last? The seasonality. Southern parts, it's for a longer period, but in the north, it's for 4 months. It's the May to September period, and it's not longer as 8 months as some of the southern states will receive. And that seasonality will also play an important role now for the biodiversity we see today. And this is the Eastern Ghats, but relatively less studied as compared to the Western Ghats and has an inverse climatic gradient. Some of the mountains here receive heavy rainfall, and as one moves north, it also gets northeast monsoon and not southwest monsoon. So has an inverse gradient. I was talking about the geological past, climatic past, contemporary climate, and as a result of it, what do we see is this extremely diverse set of habitats in the peninsular India. You have wet evergreen forest, you have the mosaic of shola grassland and a forest, you have lateritic plateaus which have an ephemeral vegetation. So vegetation or flowering is only for the few months. The rest of the time landscape looks like this. Where we are around Hyderabad, if many of you must have travelled, you see grasslands and a thorny scrub which is very similar to this. And this habitat, this complex geological past and a climatic past now holds really tremendous amount of biodiversity. And this diversity is high. We have global biodiversity hotspot, which means you have high diversity, is also highly endemic, which means it doesn't occur anywhere else in the world. It occurs only here. So high proportion of that diversity is also endemic. At the same time, it also faces threats. That's why the global biodiversity hotspots are defined. So it also faces lots of anthropogenic threats. And given this backdrop, what we are interested in understanding, okay, this is the geoclimatic past, these are the current climatic conditions. So where did this diversity, which we see so unique or endemic here, came from? Was it always there on this drifting peninsular Indian landmass? Or did it start evolving after it collided with Asia and stabilized as a landmass? Or everything else is very, very recent? In the last 1 million or 2 million years is what has it evolved. And this was the first question we started asking in our research. What are the evolutionary origins of this biodiversity we see around us? How do we do this kind of a work? And Somdata said that actually it would be nice to talk how actually the work happens. So we do extensive field work. We go and study these animals in their native habitat. How do we do that? You actually go to this forest, study them, collect the data, where do they occur, what elevation do they live in, what sort of rainfall is there, what temperature do they live in. The particular one group we study are the organisms which live in the soil, and just to give you a glimpse of it, I have a small clip here. What do we do? So this is in the rainy season when some of the animals we work with are most active. So we go looking for them. We walk through these forest patches looking for them. We turn logs, we turn rocks to collect them. And we have traveled most of the Western Ghats and the Eastern Ghats, not so much yet of the plateau, which we hope to do soon. What do we do with them? We bring them back to the lab if possible. If not, we study them in the field. We make lots of measurements on how they look, how they appear, their morphology. We also we preserve them for posterity. If not only us, others can also study them from different angle, which we haven't really explored. So we preserve them, we keep them in ethanol and in the fridge so that other people can study. This is just to give you an example. These are the collections made in 1800 by Wallace, who was thought to be a father of biogeography. So if you do this kind of a work, it's there for now hundreds of years for one person to go back and study. Once you have these animals, we also collect DNA data. And no need to say that, but DNA can capture the history of it. It tells us how the information has been passed from one generation to another generation. It has that instruction written in it saying how the individual phenotype should look like, right? And that information is held on DNA. So we use the DNA data for individual and then build their phylogenetic or evolutionary trees, which now will tell us how these two organisms are related to each other. But we are also interested in when did they evolve. So we also do something called time calibration. We use fossils and DNA together to get to these trees. So now it's not only the relationships we are aware of, we also know how old they are. And we use a lot of Bayesian and likelihood methods to do that. So a lot of computational analysis is involved in doing so. Once we have those, we then perform analysis where they may have come from. To give you an overview, imagine there is a single ancestral lineage. It has come. Every event, a new species formation is thinking about splitting event. There are two splits and it happens continuously. Some lineages, some species go extinct, some species remain with us. And this process of evolution has been going on. Now the current estimate is close to 4 billions. So you are basically reconstructing the past using DNA and fossils to figure out where are your species from, how old are they, where have they come from. And what happens, this is your present time. So you are collecting DNA here and building these evolutionary trees. And once you have that, you can see what many species, how do they look, etc. We did, and so as others in this field in peninsular India. So what sort of our current understanding is? Where does the peninsular Indian biodiversity comes from? We have biodiversity across the time scale. This is time. Sorry for the scale here, but imagine this is 0 and this goes back to 200 billion years. You have most of the organisms which are called tetrapods, like lizards, reptiles, frogs, mammals, birds are here. They are much younger. There are some arthropods which are also younger, some plants which are younger. Most of the arthropods which live in the soil are ancient. They started in peninsular India around 100 to 80 million years ago. They have been around for that long. And so has few amphibian and Sicilian lineages. Just to give a pictorial representation, this is the assemblage which came on Indian plate while it was still with Africa, Madagascar and Australia. So how do we infer this? You will have their close relatives in Africa and in Australia. So you also need DNA data from those. So it's not only you can just look at one region and do this analysis. You need your comparative data from those. So we show that many of these soil arthropod groups or the organisms which live in the soil or we call fossorial in nature, where they can go deep in the soil and live, have been around on this landscape for more than 100 million years. So they were on this drifting plate which was moving in the sea in isolation till it collided with Asia. So must have a signature now of speciation in isolation, right? We understand Darwin's theory. Where did Darwin first studied all of these things? On island. Because they are isolated, that's the unique laboratory now to study speciation. They don't have a contact with any other landmass. So imagine now your peninsular Indian landmass was traveling in isolation for close to 50 million years. You have these lineages on that landmass who are speciating. After it collided with Asia, many lineages dispersed into Asia or in Africa. Same thing happened, many lineages came into India. They dispersed from Southeast Asia to peninsular India, and all of them in peninsular India will have ages which are below 50 million years, because that's the first contact India made with Asia. After that, all of these dispersals have happened. Also, after the collision, you can also expect species come from Africa, but they are much younger, and the route of dispersal is very, very different. Right? These are the taxa where you will have the affinities in Australia, Madagascar, Seychelles or in African plate are much older. So okay, they came from these very, very different origins. They have very distinct evolutionary dates or times. But what happens when they come to this landmass? How did they actually diversify here? Do they remain as a one single species, or they became 10 or 100 or 50? To study that, we call something called diversification. And there's a small clip here. So basically this is something has been played out already, and now you are going to reconstruct that. Again, we rely heavily on mathematics here. We apply many mathematical models to understand this, but the data here is from DNA. So you have a DNA data, now you are going to use a lot of mathematical models to make inference out of it. How did this one ancestor became to 10 species or 50 species, or one remained as one and many went extinct? When we did this, what you need is this kind of a tree which is time calibrated. Of course, our research group does generate these things, but for few taxa. When we looked around, we have many such groups who are doing this kind of a work in India and abroad. We compiled 34 such groups which are unique to peninsular India, and then applied these mathematical models to understand what sort of diversification which is going on in this landscape. And this is just to give you the spread of that kind of a data. You have centipedes, scorpions, snails, frogs, birds, plants. So there's seemingly nothing common in them. They do look very different. They have come to this peninsular Indian plate at very, very different points of time. After that, how many species they have, that number is mentioned here, which is also very, very different. Now we want to see, do they have any commonality? Are they diversifying similarly or differently? How do you do that? you do something called diversification analysis. You actually measure speciation and extinction rates to understand this. And in a very simple way, what did we find? Plants are diversifying the fastest. And invertebrates, which are scorpions, centipedes, snails, are diversifying at a very slow pace. Frogs, lizards and snakes are in between. But what happens? Now we also know their age. Who is how old? Because if I am a 50 million year old group, I have 50 million years to speciate. Versus if I am a 100 million year old group, I have 100 million years. And people have hypothesized that if I am a 100 million year old group, because I have more time to speciate, I will have more species. Versus if I am a 50 million year old group, less time, less number of species. Does that hold true? And we found no. Doesn't matter how old you are, what species characteristics you have. Do you disperse well? Do you have adaptability to a newer environment? Or do you carry your ancestral niche? All of those things are determining how you diversify in a given landscape. One of the things I have mentioned here, where you come from. Do you come from Asia to peninsular India, or were you on this drifting peninsular Indian plate? Does that matter? That doesn't matter much. Who you are is, I think, one of the most significant factor which came out of our analysis, that the species traits is what we call are important, which are driving this diversification patterns. Actually, to think about it, are you diversifying with a very linear relationship, or are you showing some kind of a saturation, or there are lots of fluctuation, waxing and waning happening, or you have something like suddenly something changes and you have now 50 species. Those are called adaptive radiation. So do you have those in peninsular India? When we actually fitted these models, we said no. Most of the lineages showing gradual accumulation, which is a linear accumulation. Half of them are showing non-gradual accumulation. This has a very significant meaning to us. We talked about how the landmass was moving, climate was changing, but for certain organisms, there were always a refugia which allowed them to persist and accumulate lineages gradually, which means the build-up of the biodiversity which we see today has been gradual for most of the lineages. For some lineages, it wasn't the case, and they differed and they responded very differently to this changing climate. And again, those plots which I was showing earlier, you can actually see for individual lineage how has it accumulated. And this is the plot for all gradual accumulation of lineages. This is our present day. Their starting points are different, but the pattern they show is very similar. These are the non-gradual ones, where they are younger and the way they accumulate lineages are very, very different. And they are the ones who now are responding to the changes in paleotemperature. They are the ones who are responding to the Miocene intensification and aridification events. But what does it mean for the current biodiversity? Okay, now we have understood this is how they have diversified. How do we see them today? What sort of patterns do we see? In an area where I go to Hyderabad, outside the forest, because it's going to be a signature of all of these events. How many species will I see here? Is a product of what has happened in the past. What sort of gradients do we see in any landscape? I don't have the data for the entire peninsular India. What I'm going to show you is only for the Western Ghats. What we find, there is a decreasing trend in the diversity. You have southern parts which are much more diverse as compared to the north. Why so? Remember I mentioned this was the area which was affected by the Deccan Trap volcanic activity. Most of the biodiversity was wiped out. What's happening, that species from this area where it was a refuge are now dispersing and colonizing, and this is happening over millions of years. So what you have as a result, more diversity here, which continue to diversify there, but here you have lesser number of species. This data set is for one group of organisms called centipedes, but we did it for woody plants, for hundreds of plants, and you see the same pattern. But we also have information on something called their evolutionary relationship, right? So we looked at, are these younger or older lineages? Because if you are younger, all of them are young, then it shouldn't matter why South has more diversity. then it should be equal. If at all you are younger, you have more niches here because it's topographically and climatically much more heterogeneous as compared to the north, and also much more stable. North is much more harsher. You have a seasonality there, right? Rainfall is only for the 4 months. In the summer, it's really dry. So you don't have a wet. If you are a wet clade and you are affiliated to the wet climate, survival here is a tougher choice.
Actually, in the south, what is the selection pressure because of which this diversification is?
So the habitat heterogeneity and the climatic heterogeneity is much more higher here. So to link it to the selection pressure is for every group one has to study and make that link, but this is the more general pattern what we see. So just to wrap up, what we see that there is a graduate build up of diversity in peninsular India, but at the same time some lineages have responded to paleotemperature, Miocene climate changes, and the species trait who you are were very important for few groups. There is a diversity gradient across taxa, and this is now becoming much more common. We understand frogs, we understand lizards, we understand arthropods and plants, all of them are showing this trend. And as I said, southern has both young and old, restricted as well as widely distributed. So there are two terms evolutionary biologists like to use, something called museum and a cradle. Museum means you have old groups which are persisted for long enough, and a cradle means younger lineages are constantly accumulating. So what we have in peninsular India is both. Peninsular India acts as a museum where older lineages have persisted. They kept diversifying but at a slower pace, but you also have areas which are acting as a cradle where you have a high rate of speciation. You accumulate lineages, accumulate species at much faster pace. So you have a mosaic of museums and a cradle in this entire landscape. And this is precisely what we are trying to study, where they are. And is it similar for across groups, or is it for one group only? And the area changes from group to group. What sort of generalities are there? Or is it taxa specific, region specific, time specific? Or can we draw some more generalities? And that's where we are. And I would like to conclude this because it's not, I'm talking about it here, but it's my group and my mentors in the past and collaborators. It's a collaborative effort. So I would like to acknowledge them and multiple funding agencies who have supported this work to do this. And many, many thanks to all of you to listening to this very patiently, and to Somdatta and Tej to first of all organize this.
Yeah, I'll just invite upon Tejah to also take the dais with Jahnavi. So Tejah has a set of questions that will help us deep dive into what Jahnavi just spoke about.
Thanks everybody again, and everybody has been introducing me, so I will not waste time trying to do that again. But I just wanted to ask you a little bit, Jahnavi, about your own work and what got you interested, in biology but also centipedes.
Yes. I was interested in, I think, trekking, going outside and outdoor treks was the first introduction. But I had amazing teachers during my bachelor's and master's who introduced me to the biodiversity. I was not a evolutionary biologist or ecologist, I'm trained microbiologist. But I made a shift during my master's and then continued to work for PhD.
Yeah, no, but the bit is, it's not just biology, right? There is also this whole geography. It's a biogeography.
Yes.
That is what. So, where did you hear of it first? What got you interested in it?
So I guess during again my college days, when you are interested.
Hold it close to you, please.
Okay. Is it better? Yeah. Okay. I think going outdoors was something which was very common for me, and I think that's where I got interested in studying biology.
Sorry, where did you grow up?
I grew up in Maharashtra, in Pune, and I think going on treks was not something was very new. I did my schooling in another place called Mahad, which is the foothills of Western Ghats. So I think exposure to outdoor was very key to explore this. And when you study a biology which is in the field, you need to integrate all of these things. You need to integrate geography, you need to integrate climate to study those organisms.
Yeah, no, and this idea of, and this is something about the Deccan traps, right? I think once you start seeing them and once you see what they can offer, I think they slowly start pulling you in. I think Kobita here is also, I think, one of those who sort of really enjoys it. So, but yeah, the centipedes.
So
Centipedes.
Yes. So if you remember one of the slides where which organisms which were on this landmass while it was moving. So I was very interested in asking questions for the Gondwana biogeography. And if you really want to understand this continental drift theory, we need organisms which are equally ancient. So when I started my PhD work, these were the taxa, scorpions, spiders, centipedes and millipedes whose fossils are at least 450, 500 million year old. And that gives you a clue that, okay, these are the groups who are going to be as old as the Gondwana breakup. Because I cannot study an organism which has evolved, like for example mammals, only last 50 million years. I can't ask those questions using some of these biogeographic questions. And that's where I think, of course, one can ask with millipedes, scorpions and this thing.
That is the thing, right? So there is also this habitat. This, I think the fossorial habitat is what you had used, this idea of burrowing under the earth. So what is it about that ecological niche that, I mean, there are other, it's not just centipedes, right? There are a whole bunch of, animal life that has taken over that niche. What is it about it? Is it nutrients? Is it safety?
I think, so centipedes are not necessarily, we can't call them fossorial, they are estivate. They go inside the soil, but they also come out to live and forage and other things. There are snakes which are called shield tails, which look very different than they look like a snake, but they can go really deep and they are the fossorial. We have something called a limbless amphibian called Sicilians. Sometimes they can be fossorial. You have a frog picture, some of the frogs can go burrow. So they can have an entire life, like they just come to reproduce on the surface and then they burrow. I think stability does matter, and why, and we haven't really tasted some of these hypotheses and people are doing it now. How do you see organisms which live in the soil, the surface of soil or fossorial, are the ones we have been around for last many million years than the ones which live above ground. So people are testing some of these hypotheses. What are the traits of those organisms? How do they look at climate differently? Because temperature is different, moisture levels are different. But going back to some of your questions, why and what happens? If you have an open breathing organ, for example, you are very sensitive to the change in the climate. And many of the arthropods we study have an open breathing organ. So then you have to maintain, you have to look for a cool, wet place and to go down. So I think one of the reasons
makes sense, right? And which is perhaps why, I mean, I think in some of your work you've realized that temperature is also, and it is this idea that they're under, so they're sensitive to temperature and being under the soil sort of helps them.
Helps them also precipitation, the moisture.
Fascinating. And there's this idea of venom, right? So centipedes have venom. Why do they need venom under the ground?
Yes, so centipedes, scorpions, spiders, so all centipedes are venomous. They need their venom to capture the prey. So they paralyze the prey and then eat. They have a very well developed mouth system, so they can eat the prey, but to capture the prey, they need the venom. So they use something like called neurotoxins, which are paralytic in action. So they just paralyze the prey, so prey can't move. Once you immobilize the prey, then you can eat. So they use the venom to capture.
That sounds even worse. And so, I mean, just to sort of step back and look at all of this, right? So your work, you've been really looking at both the systematic documentation of all of this, right? So you're not just looking at centipedes per se, right? You're trying to understand where they fit in this larger ecosystem, essentially where they've come from and, and why they are where they are, right? And to me, and you're also trying to identify the processes that sort of brought them here, isn't it? That's
very, very rightly put, and I didn't touch upon that in my this talk. But one of the challenge what we have in today's peninsular Indian biodiversity is most of it's not documented. We don't know the species names, and our job starts there. We really want to understand how have they evolved, but first we need to give them a name. Like we have a name called Homo sapiens, right? We evolved 3 million years ago. So we know who is our sister species, who are our ancestors, everything. All the species which have gone extinct. That kind of a data is still happening and we are still generating for most of the organisms. So very well studied groups for this context will be birds, to the extent plants, mammals, and our majority of diversity is arthropods, which don't have names. So most of our work also goes and doing that, which is what you correctly said, systematic documentation of saying which species occurs where, and we end up describing many new species to the science, because we live in the biodiverse world, but it's not very well documented.
And so this, I think, is a good place to ask you about those basic principles, right? So what is this diversity, right? What is this evolutionary processes that achieve it, and why does it matter, right? See, I mean, we live in a city where we are striving to make everything uniform. We don't want all that change, right? So maybe do you want to look at what is look at what is evolution? Why does it matter?
Sure. So it looks like my classroom teaching, but evolution. One of the fundamental principle is called theory of natural selection, which Charles Darwin and Alfred Wallace gave us. What it is say is that in any given species you will have a variation, and that's the basis on which evolution can happen. So for example, think about any trait in humans. In this room, you may have a trait value which differs either in your skin color, eye color, height, anything. But not all traits matter for selection. There are traits which will definitely affect your fitness. And the traits which affect the fitness, selection can act upon, and then you will have a new species. So that's the theory of natural selection in a nutshell. You have to have a variation in the traits which matter, so there will be a differential fitness, and then species will survive.
Is trait everything? Is the diversity of trait everything, or is the specific trait important, right? So I may have like a longer neck. Does that matter more, or my venom is more potent?
What does a neck do is very important. So if that trait matters to a species survival, then it would matter. And that's what we call differential fitness. So if the traits you need to establish that link, that the trait matters for your differential fitness, and then that trait will matter. And selection operates on that. So natural selection doesn't drive any of these things. The variation occurs naturally in the population. Selection acts on that trait variation. And I think a very classic textbook explanation for this could be the moths, which had a variation in their wing color, right? And when Industrial Revolution happened, one of the trait which had in those butterflies or moths, which made them darker color, and that darker color had an advantage. Predators couldn't recognize them, and this black wood, the tree bark became black because of the industrial pollution, but these moths suddenly had an advantage there because they could camouflage. But that variation to have a black color wing was already in the population, which was present. Then it got selected and it could survive.
So it's the other way around, right? You should have it first,
yes,
right? You will not discover something that is not there, which is why diversity.
That's why diversity
matters. So I mean, all this is setting up context essentially, to the peninsular Indian plate as you were showing us, and what I really like was also that box and whisker chart on the side with the Gondwana and Asian lineages. Though there is a lot of, I think the horizontal line was the median, right?
Yes.
And they're basically the same despite that.
Lot of variation, yes.
What accounts for that?
That's a very good question, and I think some of my research work is in that direction. So what happens? Why, if you remember, what he's referring to, that if where you have come from, if you are an Asian taxa or a Gondwana taxa, Gondwana taxa had a very tight variation, didn't have much. All of them were diversifying in one particular way. Versus the Asian taxa had a wide variation in their diversification rates. Why so? Some of them carry their ancestral niche, where they evolved in the Asian wet tropics. And then they came to peninsular India. By the time those lineages dispersed into India, India had changed its climate. It wasn't only the wet tropics. Aridification happened, you had grasslands already coming up here, you had a savanna. So the lineages which could adapt and diversify in those landscapes, you have high variation, versus the lineages which remained and retained their ancestral niche have a less variation.
So it's kind of the other way around, right? You would intuitively, wrongly, think that the older you are, right, the more diversity you should see, but that is not what you...
See it in this data set, yes.
Ah,
We don't
of
see
course.
that. No, but people have hypothesized this, and this is one of the questions evolutionary biologists are trying to deal with, and not only for the peninsular India, but the entire tropics. It's called time for speciation. So tropics, as you saw, the Pangea are South America, Africa, Australia and Asia are the tropical areas which are closer to the equator, and North America and Europe are the temperate ones. If you look at the global biodiversity map, tropics have high diversity as compared to the temperate. And one of the explanations for that is tropics have been climatically stable as compared to the temperate regions. You may have a diversity which is equally old, but time for speciation actually is more in the tropics because of the stability.
And so here's the bit, right? So the stability is also, I mean, there is the aridification that you had talked about, but there is also the monsoon.
Yes.
Right? The variation that the monsoon brings in is also in a weird way stable.
Yes, there is a predictability.
So you had talked about it in your, I wanted to, if you could explain a little bit more about this whole, what is the relationship between the Himalayas? I mean, is India the only place where you see monsoons? That's not quite right? It's around the tropics you will find it, right? So what is the relationship?
So Himalayas actually gave the Asian monsoons, and we are the place which has this predictable monsoons. So Asian monsoons.
So what's the link, right? So is it just the gradient variation? Is it like the temperature, do you think?
No, it's the monsoon, the directionality also. We have two kinds of monsoons, right? We have southwest and northeast. And regions within peninsula receive both very differently. So you have southwest, which is from May onwards till September, and the northeast, which comes from Himalayas, is much later. And certain regions get affected differently. And the period. So where the areas receives both southwest and northeast will remain wetter throughout the year, like 8 months. Versus if the area receives only southwest, you will have only 4 months of monsoon.
And it is wet and is also refugia, I think is the word that you used, right?
Yes.
So it is also like a repository of all those old ancient species. And I just wanted to go back to that original break that you have, right? The Gondwanan. So can you explain what a taxa is? Sorry.
Okay, so species. So any group, so birds is a taxa. But within birds you will have many species. So we call those as also a taxa. So you can use loosely this term to call hornbill as a bird species, as a taxa. Amphibians is also a taxa, but a particular frog species is also a taxa.
Right. So there are all these Gondwanan species that have been here from about 150 or more years, million years or more, right? And what I think the interesting bit there is, it is these centipedes and the like that you realize are there, right? But the variation there, right? What sort of variation did you see, and why do you think that is it? I mean, what role does the refugia play in keeping them today, right? Yeah. And do you think is it like the biodiversity that whole Western Ghats are a biodiversity hotspot? But I'm sure that it is much more narrower than the whole thing, right? Is there any value in trying to protect them?
So some of our centipede work did show us that they are predators. So think about tigers and leopards, which are the predators in the carnivore world. These animals, scorpions and centipedes, are the predators in the soil world. They can also eat mice or large mammals like even bats. But they are the top predators. So predators usually are not very diverse. So you don't have 4 or 5 predators in a given area. You will have partitioning happening. So in terms of number of species, you will not find so many when you are a bigger predator. Given that we still have a really high diversity of these soil arthropod predators, of scorpions, spiders and centipedes in the Western Ghats, which tells us that much of prey, which are the other species which are also highly diverse here. So, and that diversity, as we showed in one of the maps, is not equally distributed, it's unequal. But each of the region has a unique diversity also. So, from preservation point of view, because you have a unique diversity also, I think that's what matters.
And it is like with the species, I think it is that diversity that you have to still plan for, right? And to try and preserve. Right? And also, I think there's this lovely phrase that I think that Somnatta had written about your work. I think she calls centipedes the tigers of the soil or something, right? So, sorry. There is also this question that I wanted to ask you, right? So the Asian lineages, especially the plants, they brought in like a whole new set of toolkits, right? Of these C4 plants and all. But I don't think they real. What did you see in your data set? Did you see them really take over everything? No, right?
Not really, but frankly for plants, no, we have very few groups which are well studied. Believe it or not, we have only data for 4 groups right now, and one of my PhD student is working on that, and we are generating some data. But you are correct, we don't see when you have such a, like coming from Asia, dispersing into India, you have a habitat available, but you don't see hundreds of species within every group. Why so is something which we are also trying to understand that. But if I take one more minute to clarify, some of these plants, yes, we have it from Asia, but the paleontologists in India have shown now they were also the groups which came from Gondwana, and through the Deccan traps they completely wiped out. And what you have today is actually again of a same group, similar looking plants which have come into Asia, India from Asia. So you have Cretaceous pollen, which tells you that 100 million years ago in peninsular India you had these species, then they were completely wiped out. In a similar habitat, similar requirement, similar looking species have recolonized.
Can you tell me a little bit more about, what is it about these Asian species that make them so special?
So if you, it's also something called niche conservatism, like how organisms either retain their niche or expand their niche. So many of the Asian groups which came into India would adapt to the drier environments, or they could use that environment. Just to give you an example, you are familiar with your house geckos, right? Lizards, which you see. That group came from Asia. And now you see more diversity in the drier habitats and fewer species in the wetter habitats. Took over the habitats all around Hyderabad. You go, rocky outcrops, thorny savannas, you will have more species. So it depends on which group you are and what sort of ancestral habitat you come from and how you diversify.
Perhaps it's a good time to ask you about the Deccan landscapes. Right, you've studied, as you mentioned, mostly the Western Ghats and a little bit of the Eastern Ghats. But what can you talk about the Deccan, and especially what do you see in terms of the Gondwanan taxa and the...
Yes, so frankly, before I say anything, Deccan traps as the peninsula part, even the Eastern Ghats are relatively less studied, less explored. Our understanding is just emerging now. Many of the lizards or geckos have been very well studied here. And literally every hillock has been a new species. And because if you think about it, Western Ghats is a continuous mountain chain, Eastern Ghats are much more broken. You think Deccan Peninsula is one homogeneous landscape, it's not the case. You have thorny scrub, you have dry forest, you have rocky outcrops. The barriers there for a species movement are not been reported yet. We don't understand how many species. Of course, the productivity wise it will be less. So you may not have 50 species in a given area as compared to the Western Ghats, but you will end up having a unique species. That's what the current data shows. So you may not have the species number which is very high, but what you have would be very, very evolutionarily unique because it's adapted to that particular landscape.
And it's also broken, you were saying,
much
right?
more
So
broken,
more,
it's much more
islands
continuous,
perhaps?
more islands.
This is more mathy, right? So maybe, and looking at the audience, I think there are people who will enjoy a little bit of it, not too much, I'm still here. So, but a lot of your work does require a lot of mathematics, right? You are doing a lot of,
Yeah, we are not mathematicians. We use other people's mathematics, like we use their equations to fit in our data. So we heavily rely on Bayesian approach and a likelihood approach. That's what we use it to understand our DNA, to make sense of our DNA.
What I found really interesting is that there are these tools, are open source tools that have helped a lot of other communities, right? And to see it in your work was very fascinating for me. So do you want to talk a little bit about this idea of open source work?
Yes, so open source work is like if I develop a tool, do I keep it for you have to pay to use it, or do you put the code out so that the community can use it. And most of our work, we use these open source codes, and as a community of evolutionary biologists also globally, people believe in open source code. So the program, if I write in whatever language, in Python, R or whatever, I keep the script out so that other people can also comment, other people can also use.
And what sort of work did you use? What sort of tables, what sort of these open source tools did you use?
So we use we use quite a few, and largely to deal with DNA sequence data, sequence alignments, building evolutionary trees. Once you have this time calibrated tree to fit these different models, for example, does it show a linear relationship or non-linear relationship? So to do that kind of a work we use.
And now to sort of ask you about the sort of question that you'll be hard pressed to answer, but perhaps you may want to, because of the theme of this thing, to try and sort of gaze into the future, right? What do you think you can sort of help us? What does it prepare us for, right? Your kind of work, right? You are looking at something that has happened in the past. It is bringing us to the present in a sense. What tentative steps do you think we can take into the future in an audience which is general public, so you don't have to, nobody's going to hold you, for it to the future?
No, very, very valid question, and one has to have a concrete answer, which I'm afraid I don't. But what is going to tell us is that how biodiversity has responded to the past, and can we at least broadly classify groups which will be more resilient or which will be more sensitive. And based on that, can we have any kind of a conservation management? That's where we come in. We do something called very fundamental science, but people who engage in the applied science or conservation science can then make use of our data, saying, okay, this is how the group has evolved, this is how they have responded in the past, this is where they are currently today. If, for example, a species has a very, very narrow niche, which it can tolerate of this much of temperature and this much of precipitation, we have future projections for Earth, right? We have it for 2050, we have it for 2100, and we have it for different scenarios, business as usual, if we become more worse. Now you can actually predict if a species has evolved to have this niche over 5 million years, what's going to happen when the climate is going to change. If you have a very narrow range species, your distribution is small, as well as your thermal tolerance, your precipitation tolerance is narrow, are you going to be more vulnerable? And where you are. If you are on the mountain top, there is nowhere else to go once the temperature changes. If you are a low elevation species, you are at the bottom, there is a scope to move up. But if you are already on the mountain top, what sort of future holds for you? So our data on the evolution, what their niches are going to be useful. Have we made attempts to do that? Not yet, but other people are using our data.
For now. I think I should now, thank you, that was really, now I should probably now open it up to people for questions.
Very happy to see what you're saying. Now, you have touched the subject to some extent, but I want to extend it further. The species diversity, or to that matter extinction, is a function of temperature and precipitation, is it?
Both, as well as, so some groups we see that they have responded to precipitation and temperature, paleo temperature and paleo precipitation, but some have not.
Oh, okay. Then extension of this is that going forward, looking back we have done I think enough of work, but going forward it's going to be brutal. The temperatures are going to increase like 2 degrees, 3 degrees. So every half a degree is going to wipe out certain entire species together, or did we do any modeling as what are the species that are going to survive or not?
We haven't done it, but yes, that's where one has to put in effort, and frankly we don't understand the past also very well. And I think we really need to do both, and now I think is the time to do it simultaneously, that how species have evolved in the past, and does that tell us anything how they may respond in the future. And I think one of the important factor which we don't necessarily think is the time in which we are changing the climate versus the time in which species are going to respond to it.
Yes, exactly.
If it's very fast and which is human mediated now.
And it's going to be fast.
It's going to be fast,
Very
so
fast.
then how species are going to respond to that.
Yes.
And that brings back to the question you asked, what happens if your species has more variation? it's also linked to adaptive potential. The species which are very diverse, the populations have large diversity, and diversity in terms of their genetics. They have more potential, adaptive potential to adapt. But those links and that kind of a work in India is at a very nascent stage. Across the globe, Europe is the one which is trying to do this, because Europe has already started losing some of the species which are mammals because of the melting. So, but one has to do this work, and I don't think so we are yet prepared in terms of its data, what is required to make those solid predictions. But yes.
We have Palghat Gap, which is an intersection of both east and west. I'm just curious to understand, is there something, the phenomenon of Wallace line that we see, is there something that we see here?
Very much, very much.
We do?
Yes. I don't know if you remember one of the graph which map which showed the tall mountain ranges. So across the gap, Palghat, what you have is really tall mountain ranges which go from 100 to 2000 meters. Suddenly you come down, there is a 30-kilometer wide gap, again the mountains go up. So that has been a barrier for species. And you see, and some of our work has also shown, that the communities are very distinct across the gap. So how Wallace thought about the deep marine trench as a Wallace line, as we call it, we have a Palghat Gap in peninsular India. And there will be many more. Right now we have a data for the Western Ghats to show, but in the northeast also you will have Brahmaputra, for example, acting as a barrier.
Thank you.
In one of the graphs, actually, you showed the rate of diversification is highest for the plants. So, what could be the reason for that?
Plants have something called polyploidy. So the genetic mechanism, the reproduction, they have something called polyploidy. So they go very fast. So that could be. They also have, many plants are wide ranging. So their distribution ranges are large. So these could be the potential factors which drive that higher diversification rate. And polyploidy also gives them high genetic variation.
First question, ma'am, you said that Hyderabad region is like 700 altitude something. The reason you did not mention, first thing, like it matters, why it matters, the first thing.
So what elevation we are in?
Yeah, exactly, ma'am.
So we are on a plateau. Usually we measure the elevation from mean sea level. So if I think about it, at what elevation this plateau is with respect to the sea. So we are at a 700 meter. It matters to how much rainfall you will have, what sort of temperatures you will have. That dictates all of these things.
Ma'am, and you said that most of the data is unexplored. So science is all about changing. So if we get new data, can we means the fundamental is same, but there may be a drastic change in what we think and hypothesis.
Yes.
The chances are there.
Yes. So some of the things which we call trends, where we know, then adding more data strengthen that trend. In some cases, yes, some completely unknown thing can challenge. So many of, I said, eastern Ghats or a dry savanna habitats are little less explored. So our understanding of how many species are there, how widely distributed they are, where have they come from is really limited. And if we study more, we may see that they have high diversity, but in different groups which have not been studied before. So that can change. Would it change the entire trend? May not be, but we'll have a more strength for that.
It may add to the
Yes, our understanding.
ma'am, and so any biotechnological interventions in order to increase the variation or adaptability, I mean the application part, because we have so many biotechnological means of recombinant DNA technology and all those things. So any future applications where we can fast track the process, and I know this is way back future, but asking.
Unfortunately, very, very limited, because biotechnological tools need to understand a species and a species biology really well. And something which has now, you have seen how long it takes for a species to evolve. The kind of gene editing you can do and to bring a species back is very, very, very limited. And conservation in that way is a very interdisciplinary science per se. So science has a role to it, like understanding species and species biology, but it's much more a social enterprise also. So unfortunately, biotechnological tools may not be that important to do conservation, but maybe for human health it's much more important.
Hi Jahnavi, thank you so much for the simple talk and making it easy for most of us to understand. So in Life Finds a Way and Evolution, we've been talking about so many creatures and all, but nobody asked about humans here. If you watch this series called Kala Pani, you haven't. Okay, so there is a tribe, an indigenous tribe that is that has the genetic makeup to be resistant to a particular virus. So there will be a sort of struggle between the common people and them to get the genetic material from them and use it for testing and all. So why is it that with evolution we are getting weaker and weaker, if you observe? I mean, I don't see humans being immunity-wise strong.
Think about it, I don't have a data on the human immunity and if we are becoming weaker or stronger. I'm afraid I can't answer on that. But variation, again it goes back to the variation, how much you have. And humans also have traces of Neanderthal DNA. So I think what you're describing is that we are trying to get this another tribe and get that DNA, something that humans have a chunk of DNA which is from Neanderthals. So having a DNA which makes you resistant or something, it's something we understand. But frankly, I have no clue where this human population data for the immunity is it weaker or stronger or anything.
Okay, just a follow-up. So do you think evolution is helping the human race or?
So to say that evolution doesn't help, it's a continuous process. It operates on what the variation is present. So if we are wiping out the variation in any species, it's more vulnerable to extinction. So will be humans. If you wipe out the variation in humans will be vulnerable to the extinction. So there is a theory which says if you have a small population, which means individuals which reproduce and give rise to the next generation, if anything like any disease or anything happens, you are more prone to extinction because what you are passing on to the next generations are few alleles, less diversity. So you are prone to extinction. So if we wipe out the diversity in the population, in any species, you will be more prone to variation because you will not have enough variation to fight back whatever is that disease or change in the climate or any such thing.
Thank you.
So does your study involve mycological species? Because mycology is one of the least studied things, especially when it comes to evolutionary studies. So does your study include species of mushrooms or any of these?
No, unfortunately no.
So this is more of a question to, with respect to this conservation idea, how do you people at the forefront of doing this, and also the community leaders, how do you people see this? How do we bring the change about the policy space, especially when it comes to the governance and policy space in India, particularly to India? So how do you navigate that? Do you people have anything to say about that? How do we build a case for this?
Building your own constituency is important, so as for conservationist or ecologist or evolutionary biologist per se. I guess making your information, whatever you research you do, more accessible for a policymaker, because you are not going to be the policymaker, but who is doing that job, making sure your data, your findings are available for them. And I think this is where the science communication space becomes even more important, to translate scientist work, because for us to even think and make sure our science is understandable is a task. So we do that, but then science communicators can help and making sure that it reaches policymakers. It's a dialogue. One has to engage in it, but that's it.
Thank you. You used arthropods to compare the evolution, and you said you compared this with species from Africa and Australia. What were the similarities or differences you found amongst them? And that's one question. And the other thing is, just sort of curiosity. See, if you look at these group of animals, they have not evolved so much. There are a number of species, but if you look at the way dinosaurs evolved to birds, the kind of way the way in which mammals have evolved. So why is it that certain species evolve so drastically? Of course, it's evolutionary pressure, but why is it that this group has not evolved so much in this period? It's a long time, 150, 200 million years.
So your second question, it's a very, very nice question, and it's a phenomena called stasis, which evolutionary biologists have been studying over millions of years. Certain groups look similar. They don't change their body form. As you said, dinosaurs, not within dinosaurs, but whatever, the variation was there. But certain groups don't change their body form, for example. They look similar. If I look at a 100 million year old fossil of centipedes which I study, they have a very similar body form. And we think they are under strong positive selection for that particular body form. It becomes their evolutionarily stable strategy, and that body form remains similar, persists. And there are many such examples of these living fossils is the another term people use. Yes. Yeah. So that Ginkgo biloba is one such example. Yeah. And so your first question was how do peninsular Indian centipedes relate with Australia? So you have a clades, which is like, if you think about a phylogeny in a group, you have a sister group in Australia, for example. It's also evolved in Australia. They've remained isolated, and now you have a similar number of species even in Australia. They look similar. Morphologically, they look similar. So you can actually identify you. There will be few differences, and that's what we use to identify species. But in Africa, unfortunately, very few species have been studied, but Australia is very well studied. And peninsular India is really comparable with Australia, even the climatically, because middle of the Australia is also all dry, arid habitat, and their wet forests are on the pockets. What you have, we have of course much more extensive wet forest, but Australia also is in the corners. So they show very, very similar patterns of diversification.
Just to add, is it just morphology or do you also do genetics?
Genetics, yeah.
There must be a common thread between Madagascar and the Deccan Plateau and Australia.
Madagascar and India were together for the longest time. Australia separated much earlier. So Madagascar and India separated around 90 million years ago, but Australia had already been separated 120, 130 million
There
years
is a
ago.
difference.
There is a difference between that also.
I mean, to me though, I mean, this idea of trying to think in millions of years is like, it's so hard. And I mean, the one thing that I'm thinking is almost 8. That is a time scale that I can operate in. So should we then call it an evening? Thank you so much. Thank you everybody for coming.
Thank you
so much.
for having me.
Thank you so much, Jahnavi.
Thank you so much, Dr. Jahnavi Joshi, for such an illuminating discussion and for such fascinating and important work that you do. Thanks, Tejah, for steering such an interesting discussion. Somdata, thanks so much for making this happen, and Aditya for putting the exhibition together. Thanks also to the entire GZH team and Amita for this space.
Thank you.
And maybe if I can call Kobita to give a little token of appreciation to our speakers.
Kobe didn't expect
Thank
to
you
be
so
doing
much.
this.
Thank you so much.
Me as well.
Somdata,
Thank you.
yeah. Thank you.
Thank you so much.
Thank you.
And thank you all of you for turning up on a weekday. In the words of Dr. Andrew Huberman, thank you for your interest in science.